Dear students, welcome to KPS Bio Tutorials. In the last class, we have been discussing about the formation of acetyl coenzyme A from pyruvic acid by oxidative decarboxylation of pyruvic acid. We studied that the formation of acetyl coenzyme A occurs with the help of an enzyme complex, namely pyruvate dehydrogenase complex. And this enzyme complex consists of about 3 different enzymes, and finally acetyl coenzyme A will be formed, and this acetyl coenzyme A may have different fates, and one of its fate is it enter to citric acid cycle. So today I would like to talk about in detail citric acid cycle. Citric acid cycle is a process by which acetyl coenzyme A undergoes oxidation. Here the acetyl coenzyme A undergo oxidation. In one turn of the cycle, the acetyl group with 2 carbon atoms enters as acetyl coenzyme A and eliminates 2 molecules of carbon dioxide. So this is what is happening in Kreb cycle. Acetyl coenzyme A, the acetyl part of the acetyl coenzyme A, which is having 2 carbon atoms, it enters into the citric acid cycle. By one turn of the citric acid cycle, the 2 carbon atoms of the acetyl group will be eliminated in the form of 2 carbon dioxide molecules. That means it is a complete oxidation of the acetyl coenzyme A acetyl group of the acetyl coenzyme A is taking place. The citric acid cycle or the Kreb cycle is taking place through 8 steps. 8 steps are there for citric acid cycle. The first step is the formation of citrates or formation of citric acid. The condensation of acetyl coenzyme A with oxaloacetate to form citrates. Here the acetyl coenzyme A, the acetyl part of the acetyl coenzyme A condenses with the oxaloacetate. This is the oxaloacetic acid. And the coenzyme part of the acetyl coenzyme A is released. Also one water molecules released. Then the oxaloacetic acid is converted into citrate or citric acid. The oxaloacetate is a 4 carbon compound, whereas the acetyl part is a 2 carbon compound. So this 2 carbon acetyl group will be added to oxaloacetate, thereby a 6 carbon compound, namely citrate, is formed. The standard free energy change delta G0 is minus 32.2 kJ per mole. This is the first step of citric acid cycle, and the reaction is catalyzed by the enzyme citrate synthase. Citrate synthase is catalyzing the reaction. Then the second step is the formation of isocitrates via cis-aconitic acid. The reaction is catalyzed by aconitase or aconitate hydratase. So second step is the formation of isocitrates. Here the citric acid undergo dehydration. One water molecule is eliminated from citrates by the enzyme aconitase, and an intermediate compound, namely cis-aconitate, is formed. Then to this compound, one water is added and thereby isocitrate is formed. Here, the OH group from the second from this carbon atom and the hydrogen from this carbon atom are taken together as H2O, and thereby a double bond is formed between these two carbon atoms. And again, one water is added to this intermediate compound is aconitate, and thereby the OH is added to this carbon atom and hydrogen atom is added to this carbon atom. So actually here what is happening that the position of OH and H is interchanged. Here the OH is in the middle carbon atom. Here OH comes to the fifth carbon atom. So this is the only change between citrate and isocitrate. Here the delta G0 is equal to 13.3 kilojoule per mole. The enzyme catalyzing the whole reaction is aconitase or aconitate hydratase. In first step, one water is taken away, and in second step, one water is added. Then the third step of Kreb cycle is the oxidation of isocitrate to alpha ketoglutarate and carbon dioxide. Here isocitrate is oxidized along with decarboxylation. Thereby alpha ketoglutarate is formed. So the process is a oxidative decarboxylation. So the isocitrate is a 6-carbon compound. By the elimination of one carbon dioxide, it becomes a 5-carbon compound, namely alpha ketoglutarate. Isocitrate dehydrogenase is the enzyme catalyzing this oxidative decarboxylation. There are two different forms of isocitrate dehydrogenase in all cells. One is requiring NAD as electron acceptor and other requiring NADP. Isocitrate dehydrogenase enzyme, there are two types present in all cells. One use NAD for the dehydrogenation reaction and other one use the NADP. The overall reactions are almost identical. In the case of these two enzymes, the reactions are same. In eukaryotic cell, the NAD dependent enzyme occurs in mitochondrial matrix and serve in the citric acid cycle. The citric acid cycle is taking place in the mitochondrial matrix. In eukaryotic cells, the isocitrate dehydrogenase enzyme present in the mitochondrial matrix is utilizing the NAD for the dehydrogenation. And this dehydrogenation of isocitrate into alpha ketoglutarate is taking place in 3 steps. In first step, the isocitrate will bind to the enzyme and is oxidized by NAD plus or NADP according to the kind of enzyme. Then the resulting carbonyl group sets up the molecule for the decarboxylation in the next step. So here what is happening that the NAD or NAD plus NADP will take up the hydrogen atom. One hydrogen from this OH group, other from this carbon. So the OH group attached to this carbon atom is dehydrogenated and the 2 hydrogen atoms from this carbon atom is taken away by the NAD or NADP to form NADH plus H plus or NADPH by the enzyme isocitrate dehydrogenase. Thereby an intermediate compound, namely oxalosuccinic acid or oxalosuccinate, is formed. In the second step, the interaction of the carbonyl oxygen with the bound Mn2 plus ion increases the electron withdrawing capacity of the carbonyl group and facilitates the decarboxylation. Here you can see an Mn2 plus ion. This is a part of the enzyme Isocitrate Dehydrogenase. Due to the presence of this Mn2 plus at this position, it will drag the electron. It has tendency to attract the electron from the CO group. This CO group is formed by the dehydrogenation of this carbon atom. And as the Mn2 plus pull the electrons from the CO group, the double bond is shifted, and thereby as the electrons are shifted to the oxygen from this double bond, then there will be a tendency to shift this bond to this between these two carbon atoms. So due to the shifting of the double bond, the bond between this COO group and this carbon atom will break, and as a result this COO group is eliminated as carbon dioxide. And thereby another intermediate compound is an enol compound is formed. This compound again rearrange to form alpha ketoglutaric acid. To this compound, one hydrogen ion is added to form alpha ketoglutarate. So once again I explain. In the first step, the isocitrate is dehydrogenated. Two hydrogen atoms are taken from this carbon atom. Thereby a CO group is created, and the CO group is placed near to an Mn2 plus ion in the enzyme, and thereby there is shifting of double bond. Due to the shifting of double bond, there is a tendency to break this bond between COO group and the carbon atom, and thereby the CO group is eliminated as carbon dioxide. The resulting enol compound is again protonated to form alpha ketoglutarate. So in this way, the isocitrate dehydrogenase enzyme is catalyzing the oxidative dehydrogenation of oxidative decarboxylation of isocitrate into alpha ketoglutarate. Thereby the isocitrate is a 6-carbon compound which eliminates 1 carbon as a carbon dioxide. Thereby its number of carbon atom is reduced into 5 carbons in alpha ketoglutarate. Then in the fourth step of Kreb cycle, here the oxidation of alpha ketoglutarate into succinyl coenzyme and carbon dioxide is taking place. So here also there is an oxidative decarboxylation. So in this step also the number of carbon atom is reduced. Here what is happening that the alpha ketoglutaric acid is undergoing dehydrogenation and decarboxylation to form succinyl coenzyme A. The enzyme catalyzed in this reaction is alpha ketoglutarate dehydrogenase complex. Here NAD will take up the hydrogen atom to form NADH plus H plus. This reaction, that is the oxidative decarboxylation of alpha ketoglutarate into succinyl coenzyme A, is very similar with the formation of acetyl coenzyme A from pyruvic acid that we already studied in detail. The formation of succinyl coenzyme A from alpha-ketoglutaric acid is very similar with the formation of acetyl coenzyme A from pyruvic acid by the enzyme pyruvate dehydrogenase complex. The alpha-ketoglutarate dehydrogenase complex enzyme is very closely resembling the pyruvate dehydrogenase complex in structure as well as function. It also includes three enzymes homologous to E1, E2, E3. We already studied that in PDH or pyruvate dehydrogenase complex there are three enzymes. E1, E2, E3, a pyruvate dehydrogenase enzyme is there. Then an enzyme namely lipoyl acetyl transferase enzyme was there. And a another dehydrogenase enzyme also there. E1, E2 and E3. The similar enzymes also present in the case of alpha-ketoglutarate dehydrogenase enzyme. Also, the cofactors of PDH was TPP, thiamine pyrophosphate, and lipoid, FAD, NAD, and as well as coenzyme A. All these compounds also present in the case of alpha-ketoglutarate dehydrogenase enzyme complex. So, the formation of succinyl coenzyme A from alpha-ketoglutarate is very similar to the formation of acetyl coenzyme A from pyruvic acid. So, these are the steps of formation of succinyl coenzyme A from alpha-ketoglutarate. This is the alpha-ketoglutarate. With the help of the enzyme 1, it is get transferred into thiamine pyrophosphate. By this transfer, one carbon dioxide is eliminated, and the resulting compound is called alpha hydroxy gamma carboxyl propyl thiamine pyrophosphate. Alpha hydroxy gamma carboxyl propyl thiamine pyrophosphate. Then in the next step, this compound react with a lipomide or it transfer this group into another carrier compound namely lipomide, and thereby a compound namely succinyl lipomide is formed. The succinyl group from the thiamine pyrophosphate is transferred into lipomide to form succinyl lipomide. Then in the third step, the succinyl part from the succinyl lipomide is then transferred into into coenzyme A. You know the coenzyme A is having an SH group. We have already studied in detail about this structure of coenzyme A. So the coenzyme A with the SH group take up the succinyl group from the succinyl lipomide to form succinyl coenzyme A. And thereby coenzyme A is released, and by this transfer the lipomide is reduced into a reduced form of lipomide. Then in the fourth step, this reduced form of lipomide is oxidized with the help of an NAD plus by an enzyme E3, the third enzyme. And this NAD plus take up the hydrogen atoms from the reduced form of lipomide, and thereby the lipomide is oxidized into its original form and NADH plus H plus is formed. So these reactions are very similar with the formation of acetyl coenzyme A from the pyruvic acid. Here only difference is instead of pyruvic acid, here the starting compound is alpha-ketoglutarate acid. All other reactions are same. So in this way, succinyl coenzyme A is formed after these reactions, catalyzed by alpha-ketoglutarate dehydrogenase complex. Then in the fifth step, The succinyl coenzyme A is converted into succinic acid or succinate. This is the succinyl coenzyme A. You know, this thioester bond, that means the bond between the CO group and the sulfur atom of the coenzyme A, that is the thioester bond. This bond is a very strong bond with a free energy of hydrolysis of delta G is equal to 36 kilojoule per mole. So this bond is a strong bond with high bond energy. Breakage of this bond will release an energy about 36 kilojoule per mole. So in this next step, by an catalyzed by the enzyme succinyl coenzyme A synthetase or succinic thiokinase. Succinic thiokinase enzyme catalyze the breakdown of this coenzyme A part from the succinyl coenzyme A. Thereby this bond is thioester bond is broken, and the released energy is utilized for the phosphorylation of GDP into GTP. So here one GTP is formed. And the delta G0 for this entire reaction is minus 2.9 kilojoule per mole. That means the release, most of the released energy is utilized for the synthesis of GTP. As a result, the coenzyme A is released from succinyl coenzyme A, and the succinate is, the resulting compound will be succinate. Then in the sixth step, oxidation of succinate to fumarate is taking place. The succinate formed from the succinyl coenzyme A is oxidized to fumarate by succinate dehydrogenase enzyme. The enzyme catalyzed in this step, succinate dehydrogenase. Here the hydrogen acceptase are the FAD. FAD will take up 2 hydrogen atoms from the succinate. Thereby FAD is reduced into FADH2, and here a double bond is formed, and the resulting compound is said to be fumarate, and the reaction is a reversible reaction. The delta G0 is 0, that means it is a interconvertible and reversible reaction. Now the fumarate is formed. Then the seventh step is the hydration of fumarate into malate. Here the enzyme catalyzed in this step is fumarase. The fumarate with this double bond is eliminating to this compound, to the fumarate, a water molecule is added. You know, the water molecule is added in the form of OH minus as well as H plus ion. First OH minus ion is added to this carbon atom, and thereby the double bond break, and to the valence to the single valency of this carbon atom, one H plus ion is added, and thereby a compound without double bond is formed, that is called malate. Here the delta G0 is minus 3.8 kilojoule per mole. Then in the eighth step, oxidation of malate into oxaloacetate is taking place. Here the malate is oxidized with the help of the enzyme Malate dehydrogenase. Here the NAD plus will take up 2 hydrogen atom from the second carbon atom of malate, and thereby a double bond is found between this carbon and oxygen to form a CO group, and the NAD plus is reduced into NADH plus H plus, and the resulting compound is called oxaloacetate. Malate is dehydrogenase is the enzyme catalyzing the reaction. Delta G0 for this reaction is 29.7, 7 kilojoule per mole. So in this way the oxaloacetate is regenerated. In the first step of Kreb cycle, we utilized one oxaloacetate to form citric acid. That oxaloacetate is now regenerated at the end of the Kreb cycle. So we can conclude the Kreb cycle in this manner. Here one oxaloacetic acid or oxaloacetate react with acetyl coenzyme A, or the acetyl part of the acetyl coenzyme A is condensed with oxaloacetate in the presence of the enzyme citrate synthase to form citric acid or citrate. The citrates again undergo dehydration to form a cis-aconitate. Cis-aconitate is an intermediate compound which is again hydrated into isocitrate. Then this isocitrate undergo oxidative decarboxylation. Here one carbon dioxide is eliminated and the resulting compound is alpha ketoglutarate. The enzyme catalyzed in this reaction is said to be Isocitrate Dehydrogenase. Then alpha ketoglutarate again undergo an oxidative decarboxylation, and also the resulting carbon skeleton is transferred into a coenzyme A to form succinyl coenzyme A. This reaction is catalyzed by an enzyme complex, namely alpha ketoglutarate dehydrogenase complex. Then the succinyl coenzyme A hydrolyze its coenzyme A, and the resulting energy, the released energy is trapped in the form of GTP. One GDP is phosphorylated into GTP, and the coenzyme A is released from the succinyl coenzyme A. Then the resulting carbon skeleton is called succinate. The whole reaction is catalyzed by succinyl coenzyme A synthetase. And this reaction is a substrate level phosphorylation. That means here the energy released by the hydrolysis of thioester bond is utilized for the phosphorylation of GTP into GDP into GTP. Then in the next step, the succinate is again undergo dehydrogenation. And here the hydrogen acceptors are the FAD. FAD is then reduced into FADH2. The enzyme catalyzing this dehydrogenation is succinate dehydrogenase, and as a result the compound namely fumarate is formed. Fumarate again undergo hydration by the enzyme fumarase to form malate. Fumarate is a compound with a double bond, whereas malate is without any double bond. Then malates again undergo dehydrogenation to form oxaloacetate, and here the hydrogen acceptors are NADH. So by one turn of the Kreb cycle, 3 NADH molecules are formed. Now from the step 3, 4 as well as 8 NADH molecules are formed. And from the 6th step, one FADH2 is formed. Also from the 5th step, one GTP is formed. As well as 2 carbon dioxide is eliminated. From the step 3 as well as 4, totally 2 carbon dioxide is eliminated. That means here the incoming compound or incoming group is the acetyl part of the acetyl coenzyme A. The acetyl part is having 2 carbon atoms. These 2 carbon atoms are eliminated in the form of carbon dioxide. So this is the net reaction of Kreb cycle. I hope all of you understood the topic very well. Thank you for listening.